Soil-borne Rhizoctonia species cause major yield losses in crops like tobacco, tomato, soybean, and corn. Developing effective disease management strategies requires a uniform, viable source of pathogen inoculum. This protocol provides a standardized visual guide for producing Rhizoctonia solani inoculum on sorghum seeds. Precision is fundamental to ensuring the reproducibility of this protocol. Begin by cleaning your digital laboratory balance and placing your clean weighing container onto the pan and press the tare to prevent cumulative measurement errors that could alter your final moisture to seed ratios. Weigh 174 grams of sorghum seeds to establish a precise seed to water ratio, ensuring consistent and uniform moisture absorption throughout the entire batch and reproducibility across experiments. Next, transfer the 174 grams of weighed sorghum seeds into 1 liter Pyrex storage bottle. Add 125 milliliters of sterile distilled water. Gently mix the seeds and water. This standardized mass and volume serve as the baseline for achieving optimal hydration conditions. With the cap loosened, place the Pyrex bottle into the autoclave and run the first sterilization cycle at 121 degrees Celsius and 15 psi for 60 minutes. This initial cycle eliminates vegetative microorganisms, including bacteria, yeasts, and non-spore forming molds. Immediately after removing the bottle from the autoclave, mark the date on the bottle label. This simple but critical step prevents confusion during the multi-day sterilization timeline and ensures that the substrate completes the exact required autoclaving sequence before inoculation. Allow the bottle to cool down to hold and shake the sorghum seeds vigorously. Shaking after every single autoclave cycle is vital to redistribute moisture evenly and break up clumping. To ensure a completely sterile substrate, repeat the autoclaving process for a total of 3 consecutive cycles. All inoculation steps must be performed inside a biosafety cabinet or laminar flow hood to maintain sterile conditions. Using a sterilized 5 mm diameter cork borer, cut exactly 20 plugs from the actively growing colony margin. Use a sterilized inoculation needle to carefully lift each plug and transfer it into the prepared sorghum bottle. Distribute the 20 plugs evenly across the top surface of the seeds to promote uniform colonization. Immediately after transferring the final plug, loosely secure the bottle lid to allow gas exchange during incubation. Incubate at room temperature for 19 days. Shaking thoroughly every 4 days to break up seed clusters and ensure uniform Rhizoctonia solani colonization throughout the batch. During incubation, periodically mix the bottle by incorporating a combined shaking, rotating, and rolling motion every 4 days. This mixing technique ensures uniform distribution of colonized grains throughout the entire substrate mass and serves a dual purpose. It mechanically breaks up early grain clusters from mycelial binding causes It mechanically breaks up early grain clusters before mycelial binding causes permanent clumping, and it prevents anaerobic conditions from developing in the lower layers of the substrate that could inhibit fungal growth. Following the 19-day incubation period, the sorghum seeds should be completely enveloped in dense, healthy mycelial growth. To verify the purity and viability of the inoculum, a critical quality control step is performed inside a biosafety cabinet or laminar flow hood. Using sterilized forceps, randomly select 6 colonized seeds and transfer them onto a fresh potato dextrose agar (PDA) plate. Incubate the plate at room temperature for 24 hours. After 24 hours, observe fresh fungal growth emerging from the plated sorghum seeds, confirming successful colonization and inoculum viability. With the verified inoculum prepared, the growth chamber experiment setup was done. 12 Rhizoctonia solani colonized sorghum seeds were placed on top of the base layer in an equally spaced arrangement. Uniform distribution of inoculum. Finally, apply a top layer of the wetted starting mix to completely cover the inoculum. By day 28, the seedling growth period concludes and the harvesting phase begins. To preserve the intact root structure for accurate growth and biomass measurements, scissors are used to carefully cut open the base of each plastic cell. The seedling out of the stem should be avoided. Cutting the plastic container allows the cell plug to be removed intact without breaking or damaging the fine root network. Next, submerge the root system into a large 20.5 quart storage container filled with clean tap water. Gently swirl the roots below the surface to wash away all remaining starting mix particles. This submergence technique cleans the roots thoroughly while preventing tissue damage that can occur under direct running water pressure. Immediately after washing, place each seedling flat onto a clean paper tissue. Transfer the intact seedling directly to the digital balance to record the true fresh total weight. Next, transfer the seedling to a clean cutting surface and use a sharp precision cutter to separate the leaves from the roots. Weigh the fresh leaf and root portions separately and record each weight individually. All measurements are recorded in a laboratory notebook as a primary data backup and transferred into an Excel spreadsheet for organization and statistical analysis.